So now we are moving on finally to Cheng'En Xi, who is a second year health sciences student at McMaster University. Currently, he is interested in learning more about the COVID 19 pandemic, including its medical aspect, the social and economic impacts, and the measures we are taking to beat it. Here he is with the current trial results of SARS CoV 2 vaccines review. Thank you.
Hello everyone, my name is Cheng'En Xi, and today I will be discussing my presentation titled Current Trial Results of SARS-CoV-2 Vaccines, a Review. The presentation will follow the order that you see on this table of contents. I will first talk a bit about COVID-19 itself, and then go into the details of each vaccine trial. The COVID-19 outbreak initially emerged in Wuhan, China in December 2019. Later in March, the World Health Organization declared it to be a global pandemic. As of August 26, it has spread to almost every country in the world, leading to more than 24 million cases and over 820,000 deaths worldwide. The virus causing the outbreak, SARS-CoV-2, was first reported to be from the Huainan seafood market in Wuhan, but later studies have demonstrated more than 45% of the early patients before January 1st were not linked to this market, which further questions the origin of this virus. Animals such as bats, minks, and bamboo rats have all been speculated as potential intermediate hosts of the virus. However, there is yet to be any conclusive evidence, so that the origins of the virus is still under investigation by the scientific community. Fun fact, the picture of the bat on this slide is actually a human-sized bat found in the Philippines. SARS-CoV-2 is a novel coronavirus and is the third zoonotic coronavirus disease after SARS and MERS. It is an RNA virus with an 88 to 89% similarity to two SARS-like coronaviruses derived from bats. It also shares a 79% similarity to the SARS virus and 50% similarity to the MERS virus. An early study estimated the R0 value to be 2.2, meaning each infected individual can infect 2.2 people on average. SARS-CoV-2 has spike proteins that give it its distinct crown feature, hence coronavirus, and play an important role in binding to receptors on host cells. The spike protein interacts with epithelial cells in the human respiratory system by the ACE2 receptor. The protein contains an S1 subunit that has a receptor binding domain, or RBD, which allows for the binding of virions to host cells. After the S1 RBD binds to the ACE2 receptor, the S2 subunit inserts itself into the host cell and mediates membrane fusion. Neutralizing antibody and T-cell responses are used to measure immune responses. Antibodies are proteins that attach to foreign substances for the immune system to sense and eliminate, but only a small subset of antibodies are neutralizing antibodies. These neutralizing antibodies bind to viruses in a way that inhibits infection. One way to achieve this is through the blocking of virus interaction with the receptor of the target cell. In addition to antibodies, T-cell responses are crucial to the long-lasting immune response against viral infections. To quantify the antibody response, a titer is used to determine the concentration of antibodies in the blood. The geometric mean titer, GMT, is typically the standard to determine the average antibody response in a group of subjects. Another measure of antibody response is seroconversion, which is the period during which antibodies become detectable in the blood. The objective of this review was to examine and summarize the most recent published human or animal clinical trial results of COVID-19 vaccines, focusing on the methods, results, and discussions of each published study. A list of all COVID-19 vaccine candidates currently in development was obtained through the Government of Canada website. All vaccine candidates undergoing testing were searched and identified using PubMed to obtain any published data from their studies. If a vaccine candidate has multiple studies published, the one with the biggest sample size is selected, and human trial reports were preferred over animal models. All numerical data included has a p-value of less than 0.05. The first vaccine candidate I'll be discussing is the ChAdOx1 nCoV-19. It is developed by the University of Oxford and AstraZeneca. It consists of the replication-deficient simian adenovirus vector, ChAdOx1, while containing the spike protein of SARS-CoV-2. This was a phase 1 and 2 single-blinded randomized controlled trial study performed at 5 different testing centers in the UK. Participants were between the ages of 18 and 55, and individuals who had been infected with this virus already, or people who are at a high risk of exposure were excluded. They were randomly assigned to receive either the experimental vaccine or the MenACWY, which is a placebo. The placebo will lead to a common local or systemic reactions of viral vector vaccination, which would maintain participant blinding. Participants were separated into 4 groups. Group 1 acted as the phase 1 component of the study and had intensive follow-ups. Group 2 was assessed for both humoral and cellular immunogenicity, while Group 4 was only assessed for the humoral component. Group 3 consisted of a non-randomized group that received the booster shot 28 days after their first dose. Group 3 participants were not blinded and had only 10 people. Within groups 1, 2, and 4, they were further divided into the experimental group or the control group. Prophylactic paracetamol was given to 56 participants in the ChAdOx1 group and 57 in the MenACWY group. This reduced the incidence of adverse events such as pain, fatigue, headache, and fever. The use of paracetamol did not affect the immune response, and the incidence rates of these events among these groups can be seen on this table. After 1 dose, antibodies targeting the spike protein peaked by day 28, and among the 10 participants who received the booster shot, the median antibody concentration on day 56 further increased. On day 28, all participants achieved neutralizing titers. It was also found that the titers inducing 80% virus neutralization was achieved in 91% of the participants after just 1 dose, and in 100% of the participants following the booster dose. In addition, just 1 dose of the vaccine candidate resulted in 62% of the participants having neutralizing antibodies that completely inhibited the cytopathic effect of SARS-CoV-2. The same occurred in all participants after a booster dose. The findings showed that ChAdOx1 nCoV-19 was safe and tolerated in human participants, while inducing strong antibody responses, including neutralizing antibody responses. The majority of adverse events reported were either mild or moderate in severity, and all were self-limiting. The use of prophylactic paracetamol increased tolerability while not impacting immunogenicity, which can help reduce the confusion between vaccine-related symptoms and actual COVID-19 symptoms. The limitations of this study include the short follow-up reported to date, the small sample size in the booster group, the lack of overall diversity in the sample population, and the single-blinded design. These are being resolved in phase 3 trials, which have begun in countries such as South Africa. The second vaccine candidate being discussed is the 85 nCoV, developed by CanSino Biologics in China. Similar to ChAdOx1, it uses an adenovirus vector that expresses the SARS-CoV-2 spike protein. The participants received either a medium or low dosage or a placebo shot as intramuscular injections. This study was randomized, double-blind, and placebo-controlled, and conducted at a single testing center in Wuhan, involving healthy adult participants with no upper age limit. No one with previous SARS-CoV-2 infections or serious chronic diseases were selected. In total, 508 participants were involved in the trial. Within 14 days post-vaccination, adverse reactions were reported by 72% of the participants in the medium dose group and 74% of the participants in the low dose group, significantly higher than the placebo group. The most common adverse reactions were pain at the injection site, fatigue, fever, and headache. You can see the incidence rates in this table. While most were reported as either mild or moderate, 9% of the participants in the medium group had severe reactions, but these reactions ended within 72 to 96 hours without the need for medication. On day 28, more than 96% of the participants in both dose groups shows seroconversion of the RBD-specific antibodies. Seroconversion of neutralizing antibodies only occurred in about 58% of the participants receiving the medium dose, and in only 47% of the participants receiving the low dose after 28 days. Age greater than 55 and previous exposure to immunity to type 5 adenovirus were found to have a negative impact on the antibody responses. Despite these factors, all antibody responses remain much higher than the placebo group. Significant spike glycoprotein-specific T-cell responses were induced in more than 87% of the participants in both dose groups. No immune responses were observed in the placebo group. The data showed that a single injection of the AD5-nCoV at either doses could induce spike protein-specific antibodies, though with lukewarm neutralizing antibody responses. The vaccine candidate also showed a good safety profile, with most reactions being mild or moderate. The limitations include the lack of diversity in participants, the lack of data past 28 days post-vaccination, and the lack of live SARS-CoV-2 exposure post-vaccination to assess efficacy. The limitations emphasize the importance of an international, multi-centered phase 3 efficacy trial. The final vaccine candidate discussed is the mRNA 1273, developed by Moderna. Unlike the previous two, it is an mRNA vaccine that encodes an antigen to the S2P site of the virus. It is also the only candidate reviewed to not have any published phase 2 trial reports as of now. All participants were also given 2 injections 28 days apart, also unlike the previous two. This study was a phase 1 trial that only involved 45 participants in total, with 15 in each of the 3 dose groups. Participants were between the age of 18 and 55 and were instructed to not routinely use analgesics or antipyretics before or after the vaccinations. Initially, all adverse effects were reported as mild or moderate in severity, with incidence rates increasing after the second vaccination. Adverse events occurred 54% of the time in the 25 microgram group, and every participant in the 100 and 250 microgram groups after the second injection, with 3 participants reporting 1 or more severe events. The most common adverse events across both vaccinations included fatigue, chills, headache, myalgia, and pain at the injection site. Binding antibodies to the S2P site of the virus and RBD-specific antibodies increased rapidly after the first vaccination, and seroconversion occurred in all participants by day 15. After the second vaccination, neutralizing antibody responses were detected in serum samples from all participants. Moreover, at day 43, wild-type virus neutralizing activity capable of reducing infectivity by 80% or more was detected in all participants, with the average responses being above the convalescent serum specimen tested from COVID patients. All immune responses were dose-dependent. mRNA 1273 was able to induce a strong immune response in all participants after 2 doses of the vaccine, while showing limited adverse effects. The limitations of this study include a very small sample size, a lack of ethnically diverse samples, and the lack of a placebo control. A phase 2 trial of this vaccine candidate evaluating doses of 50 and 100 micrograms is ongoing, with a large phase 3 efficacy trial expected to begin during this summer, which will hopefully resolve those limitations. In conclusion, all 3 experimental vaccine candidates reviewed have shown strong immunogenicity and safety profiles in their respective phase 1 and 2 human trials. All the 3 vaccine candidates are expected to conduct their respective phase 2 or 3 clinical trials as soon as possible, as well as following up with the participants involved in their published trials. A special thanks to the Antarctic Institute of Canada for making this presentation possible, as well as a special thanks to Austin Martin, Peter Anto Johnson, and John Christie Johnson for their contributions to the research article. Thank you very much. And these are my references.
Awesome. Thank you very much, Cheng'en. The next question goes out to Cheng'en. I have seen people debate about accelerating trials. Is this being done, and can it be done safely?
To answer the first part of this question, is it being done? The answer is yes. But The idea of accelerating trials usually currently that is being done is that instead of having several separate trials, you will have several trials that's mixed together. For example, the Oxford vaccine that I just discussed, ChAdOx1, it was a phase 1 and 2 study, with group 1 being the phase 1 component. So what they did essentially was have the phase 1 component happening around at the same time as the phase 2 component, and they are planning to have a phase 2 and 3. So that is their way of accelerating trials, not necessarily having less follow-up time, as they do plan on following up with their participants later on. Additionally, the 85 nCoV vaccine from China, the phase 2 study that I just discussed, the phase 2 study actually began before all the data from phase 1 was in and all the data was processed. And their study actually specifically mentioned this as another one of their limitations. So yes, this thing, accelerating trials, at least in different ways, is totally being done. Now, as for can it be done safely? So far, we haven't, as at least for these three vaccine candidates, as those are the only ones I have extensively studied. They all show a very strong safety profile, and so far in their trials, nothing dangerous has came up, and all the adverse events were self-limiting. Everything was limited. So can it be done safely? The answer is most likely yes, but it has to be. I believe it has to be judged on a vaccine by vaccine basis. These vaccines have published data, and not just a single published data. They have also conducted previous trials on animals or phase 1 studies. So if the scientists developing the vaccine feel confident about accelerating trials by looking at the safety profile of their previous data and by having some degree of restraints in accelerating the trials, then I believe there is the chance of conducting it safely and effectively.
Awesome, great comprehensive answer. So we don't really have too many more questions. I did have actually a question for Cheng'en though. Out of the candidates that you took a look at and you reviewed and analyzed a little bit, do you think that there's one that's in the lead to become sort of the first vaccine that countries actually adopt? And how do you think that'll go?
That is not an easy question to answer. For example, as I'm sure many of you have seen the news, Russia actually just approved the vaccine without releasing any testing data. So, can they approve things? Yes, obviously, but it comes to the problem of whether or not you're approving it with all the necessarily all the necessary tests being done. Now, as for in the lead, currently I know that the Moderna vaccine is conducting a phase 3 trial. The same thing is happening with the other two vaccines. The only difference being that the among the three vaccine candidates that I have looked at, the Chinese one, 85 nCoV, they do somewhat poorly in terms of neutralizing antibody responses, although they don't have any safety concerns. However, this vaccine actually just received a patent from the Chinese government to become the first COVID-19 vaccine actually patented by the country, even though it has not entered the market, and and China has approved it for military usage only for 1 year. So have they approved it? Approved it? Yes, but can you really consider to be in the lead? Not necessarily. Currently, all three of these vaccines are considered front runners in their respective countries as well as in the world. But at a certain point, it does not necessarily matter if your vaccine is going to come out a month or 2 months before the other vaccine. If you're not following the safety procedures and potentially feed into all the anti-vaccine sentiments or risk causing physical or bodily harm to whoever takes the vaccine.
Okay, thank you very much, Chang'en. Another thank you to the presenters, all of you today who